Today, we are privileged to have our scientific director, Pamela Feliciano, with us speaking about genetics and autism. Pamela Feliciano is Spark's scientific director and a senior scientist with the Simons Foundation Autism Research Initiative, SAFARI. As a senior scientist with SAFARI, Feliciano is deeply involved in developing and managing several projects whose missions are to build research cohorts of individuals with autism. She also works on developing objective and reliable outcome measures for autism clinical trials. Previously, Feliciano worked as a senior editor at Nature Genetics, where she was responsible for managing the peer review process for research publications in all areas of genetics. She has a bachelor's degree from Cornell University, a master's from New York University, and a Ph.D. in developmental biology from Stanford University. Pamela also has a son with autism. It's our extreme pleasure to have Dr. Feliciano with us today, so without further ado, take it away.
Great. Thanks, Vincent. I'm Pam Feliciano, and I'm really happy to be here today to talk about the role of genetics in autism. So today's talk will focus on, well, first we'll talk about my personal connection to autism. Then I'd like to start with a big picture and talk about why genetic research is important in autism. Then we'll spend a little time on the nitty-gritty details of DNA, genes, and chromosomes. And then we'll end by talking about Spark and how Spark is working with lots of families and individuals with autism to power more research. So in 2004, I finished my graduate degree, and I was working as a scientist. And in 2007, my son, who was 2.5 at the time, was diagnosed with ASD. And on that day, I had many WH questions for the physician who gave the diagnosis. What can we do? Where can we get help? Who can help us? And what I learned in that appointment meeting is that quality autism treatments are really difficult to find, and they're difficult to fund. And when I asked why, what happened, why does my child have ASD, there were really no answers. At that time, this was 10 years ago, the doctor felt like she couldn't tell me anything about the reason why my son had this diagnosis. And I think at Spark, what we recognize is that all families have questions about their own individual family member or themselves, and so everyone is unique. And what Spark is doing is partnering with the whole community to accelerate research on lots of topics. So, go ahead. Maybe I should. All right, so let's start with the big picture and talk about how researching genes helps people. Okay, so I think it's important to understand that the major thing that genetic research does is increase our knowledge about the biology of autism. So it helps us understand things like, helps us understand how these genes work in the brain, and it gives us some insight into how the brain develops and functions. It also helps us understand how genes are connected to changes in behavior. It helps researchers do better, more focused research when we have an understanding of the genes that are involved. And it may help us understand why there are some comorbid medical conditions like ADHD, epilepsy, sleep problems, seizures, why they occur in some individuals with autism but not others. And finally, what genes do is they give us the building blocks, the foundation that we need to develop more effective treatments and therapies for individuals with autism. I think at an individual level, genetic research can also help a family or an individual affected with autism. In Spark, we've identified a genetic result in 6% of families so far, and we expect that number to grow over time. So in 3 or 4 years, I think we could expect that number to double, or maybe even more than that. And if you get a genetic diagnosis, it's really helpful to that family because it ends the journey of not knowing what caused the person's condition. So you have a better understanding of that person's condition. You can understand the impact of the risk of autism or the condition in other family members, or the children of other family members. You can get a better understanding of what the future might hold, because even though these genetic conditions only occur even though we only are able to return results in a small percentage of families, there still are, I'd say, tens or hundreds of people with genetic diagnoses, and there'll be people with different ages. So you'll get a better understanding of what the future might look like. You also will get an opportunity to network with participants with the same genetic diagnosis, which is very helpful to share tips and talk to each other. And finally, you'll learn about trials or treatments specific to your genetic diagnosis in the future. So there are lots of benefits that genetic research has for the whole community as well as individual families. So let's talk a little bit about the nitty-gritty of DNA and autism genes. I think it's important to understand that we've known for a long time that autism is both genetic and environmental, and we've learned this through twin studies. So if you look at identical twins and one has autism, the chance that the other one has autism is very, very high. So 77% of identical twins, if one has autism, the other one does also. If you look at fraternal twins, which share the same prenatal environment, but don't share the same genetics, same genes as identical twins, there's a high concordance. There's a concordance rate of 31%, which is actually higher than siblings. And so siblings are just like fraternal twins in terms of their genetics, so they share the same amount of genetic information. But you'll see that because the fraternal twins share the prenatal environment, they have a higher rate of concordance. And what that tells us is that there is also an environmental component to autism risk. We've learned a tremendous amount about the details of the risk factors for autism. So we know that genetic factors play a really large role in autism. Somewhere maybe 50 to 80% of autism can be explained by genetic factors. We also know, again, that there's environmental factors, and it's estimated that perhaps that's 20% of autism, and as well as there's a combination. We've learned through genetic studies a number of autism genes, so I would say that we understand, we've identified 100 autism genes. There are probably several hundred more that contribute to autism risk. But we have learned a lot in the past 10 years about the genes that lead to autism. Okay, so what is DNA and what is a gene? So DNA is a molecule that's present in all of your cells that provide instructions for your body. So if you see on this diagram here, you can see what we're representing is that genes encode proteins. So this red protein is a protein that's specific, that's necessary in one's brain. The blue protein is something that's necessary in lungs, and the green is just represented as necessary in the colon. And what this means is that each gene is present in every cell, and the genes encode proteins which are needed for each of your tissues to function properly. The genes are encoded in the genome. So the genome, when someone says genome, they just mean all of the genes that you have. And the genome is broken up into chromosomes. And so the chromosomes have genes on them. Okay. So genes also have sections called exons and introns. So maybe you've heard the term exome. The exome just means the parts of the genome, parts of genes that code for the protein. So the exons are the actual part of the DNA that give the instructions on how to make the protein. The introns are not used for the structure of the protein, so it's just, I guess you could think of it as empty space. So the exons are the most understood part of the genome, and that's what we sequence in Spark. So if you hear the term exome sequencing, it means that we're sequencing all of your exons. Okay, so let's talk a little bit about inheritance. So as I said earlier, all of your genes are called your genome, and most of these genes that you get are inherited as exact copies from your parents. So you get 23 chromosomes from your mom and 23 chromosomes from your dad, and that makes up your genome. And there are 20,000 genes, and there's 20,000 genes in 23 chromosomes from dad and 20,000 genes from mom, and so you have 2 copies of each gene. And most of the time, the genes that you get are copies from your parents. And what's important to note that sometimes there can be changes, and these types of changes that are not inherited from your parents actually turn out to be really important in autism. So we know that probably a quarter of autism may be explained by these de novo genetic changes. And so you'll see here that the genetic change is not present in the mom or the dad. It just occurred in either the egg from the mom or the sperm from the dad, and then is therefore present only in the child with autism. Okay, there are other types of genetic changes that can lead to autism. One such change is something that's called an X-linked mutation or X-linked change. This diagram shows how that works. So typically, the mom will carry a change in one of her X chromosomes, and if she has a son that happens to inherit that particular X chromosome, that boy may have autism because that child doesn't have another copy of the X chromosome. She can also pass that chromosome on to a female child, and most of the time the female child won't have symptoms of autism because she will have another X that doesn't have the genetic change that leads to autism. There are other aspects of genetic changes that I think that are important to understand. So in terms of size, a genetic change can be small or big. So an example of a small genetic change would be something that changes one tiny part of a protein. It can just change what that little part looks like and what it is, and it will disrupt the function of that protein. These types of changes can have big impacts, or they can have small impacts, even though it's small size, but we'll get into that later. In terms of size, again, there can also be big changes where there's a whole part of a chromosome either missing or duplicated. And I think the take-home message here is that you can have small or big changes, and that these changes can have different impacts. Usually a big change means there's a big impact. Sometimes a small change, like a missense mutation, can have a big impact. So the size of the change doesn't always correlate with the effect size, but I think by and large, for the most part, if there's a big change on a chromosome, it is likely to have an impact. Okay, it's also important to know that some genetic changes have strong effects and they're rare. So here we've sort of diagrammed a population of individuals, and these 2 green people are just representative of people who carry these very rare genetic changes and they have autism. And as I was saying before, we've actually learned a lot about the genes that lead to autism, and so we've discovered, researchers in total have discovered about 100 genes that have small changes that almost always cause autism. But yet there are still several hundred more that we need to discover. There are also changes that have small impact and also play a part in autism risk. So some people with autism have these so-called common variants. So if you just look at this diagram, what we're showing here is that all the people with the DNA in their hands have autism linked common variants, but only some of them will have the actual autism condition. So far, researchers have only discovered one common variant that's associated with autism in this way, and we know that there are probably hundreds, maybe thousands of these variants, and we need to study many, many more people with autism to discover the rest of these variants. So to summarize and look at the risk factors for autism overall, what we've talked about is that there are environmental factors that really aren't well understood and estimated to contribute to 20% of autism risk. The rest is genetic, so we can estimate that there's about 80% of risk in autism is due to genetic variants. A quarter of those genetic factors are the so-called de novo mutations. These are the ones that are only found in the person with autism and not the parents. And then there are inherited variants and also somatic variants, which we won't talk about today. I should say that we understand the most about these de novo variants, and that's just because when you find a mutation that's rare in the general population and only present in autism, you have a good sense, a good understanding of whether that thing is likely to cause autism, and it is. These other types of variants are definitely things that we hope over the next 5 to 10 years we understand better through analyses that we do at Spark and other researchers. But for now, we'll mostly focus on de novo mutations. Okay, so we'll talk about now how Spark is working together to find answers in autism and what we're doing. Okay, so as you all know, autism is quite variable in how it presents. So the symptoms of autism are very different in different people. And also, as we've shown, the causes of autism are very different in different people. And what we need, what researchers need, is a way to subgroup individuals in a meaningful way so that we can have more reproducible and better research. Because what's happening now is that researchers recruit a group of people with an autism diagnosis and then do research, and then it can't be reproduced because the group of people that they started with was so variable. And so what we hope we can accomplish in Spark is provide that to researchers. So what we're doing is recruiting 50,000 families and collecting information from them, including their genetic data if they consent to it. And we're returning genetic results so far only to 6%, but we do feel that that number will definitely increase over the next 3 to 4 years. Returning genetic results so that participants can be grouped by these genetic similarities and be matched to research studies that are studying their particular diagnosis. And in that way, we'll be able to power better research and more reproducible work. And so in terms of who can join, anyone living in the United States with an autism diagnosis and can join Spark. We also invite first-degree family members, so parents of the child or adult with autism are welcome to join, as well as biological siblings. Participation is relatively simple and can be done from home. So everything can be done online. You just need to log in to sparkforautism.org, invite your family members, and also return your saliva kit to us, and you'll hear about other research opportunities from us. So far, we've recruited 100,000 participants in Spark, and this map shows the density distribution across the country. And the orange dots are our different clinical sites that we're working with to recruit most of the participants. We've recruited 36,000 people with autism, and so far we've gotten DNA from 10,000 families. Okay, so the one thing that I wanted to highlight here for you is that how we know that these de novo variants are really important in autism. So what we've done in Spark is to look at all the de novo genetic variants that are present in the people that we sequenced. And when we compare them to people without autism, we see that people with autism have an enrichment of these gene-disrupting de novo variants. They have a twofold enrichment over people without autism. And what that tells us is that there is a signal here. Because people with autism have an enrichment of these types of changes, we know that these types of changes are a strong indicator, a strong genetic factor in autism. If you look at these small changes that I talked about, the missense changes where the effect is relatively, can be weak in some cases, you see that we have somewhat of an enrichment in Spark. So it's 1.2 times more common in people with autism versus people without autism. And what that means is that some of these missense changes lead to autism, but not all of them. And then an important control to look at here is whether de novo changes. So as I was saying earlier, de novo changes can lead to a major change in the gene, or the change can not have any impact if it's silent. So that's a change that wouldn't change the gene. And you can see here that there isn't an enrichment in people with autism. So people with autism have the same number of silent de novo genetic variants as people without. And so that's a good control to see, because you wouldn't expect those changes to cause autism. And again, looking at all of these numbers, we know and we can see that these gene-disrupting de novo variants are contributing to autism and can explain autism in some people. And so here's just another visual way of looking at that. What we do in Spark is we sequence all the people with autism and their family members. We identify the de novo gene-disrupting changes only in the people with autism, and that's here in the blue circle. And then we remove all the changes that are seen in people without autism. And by that way, we can identify the variants that are really important and can likely lead to autism. So hopefully that was clear. So I just wanted to go over that one more time. So what we're doing is we're looking at changes in autism and then taking the variants that are present in the regular population and removing those, and then we focus on this piece here on the left, the variants that are only present in the people with autism. Okay, so doing that, we've identified so far 6% of individuals with autism in Spark that have a returnable result. So lots of these genes have been found before in autism, and we're starting to discover new ones. We're not returning those results just yet, but over time we will, because the numbers will increase and then we'll have more people and better statistics of whether that gene is involved in autism or not. Okay. And I think what's important here is that we started to return results to families, and if you stay tuned to Spark, you'll start to see some of their stories on our Facebook and Instagram. And so far, 6% have gotten a result, and we expect that number to grow over time as we learn more. So if we just come back to this diagnosis day slide, I think that Spark is definitely going to power more research and get to better answers. It is gonna take time, and we definitely hope that you'll join us and be part of our journey. And I have a lot of people to thank, and also just mention, if you haven't joined, you can visit us at sparkforautism.org, tell others about Spark, and we want to just thank all of the individuals with autism and their family members who've joined and have helped Spark understand how we can better power autism research.
Thank you, Pam. So we received a number of questions. You can continue to send us questions through the chat box. So let me just sort through some of these here and ask them aloud. Can you re-explain X-linked changes? Someone just wanted a little more clarity around is the ASD gene in the X chromosome?
Yes. So I should have clarified this earlier. So there are probably 500 to 1,000 genes on every chromosome in the genome that are linked to autism. Autism is very different than other conditions like Huntington's disease or cystic fibrosis, in that there's only one major gene associated with the condition. So autism genes can be present on everywhere in the genome, and so they can be present on the X, they can be present on chromosomes 1, 2, 3, 4, 5, and if they are present on the X, they oftentimes are carried by moms who aren't affected, but then if they pass that chromosome on to a male child, that male will be affected.
So you mentioned 100 genes, or even more than 100. How many are on our list right now?
Right now it's 90, but we just had a meeting the other week where we added 20 more. So we have to, it's about 100.
Okay.
Yeah, 100, 110.
Someone is asking about environmental factors.
Yes.
Being prenatal or after being born.
I think the ones that are best understood are prenatal. There have been a number of studies. Well, the strongest factors that are known are exposure to valproic acid. So this is a medicine a mom would take for epilepsy. That's a clear risk factor. Less strong evidence for things like fever in pregnancy. There also have been 2 or 3 studies showing an effect of proximity to air pollution as being a risk factor for autism. But none of these environmental risk factors have the strength of an impact of, say, a de novo mutation in a bona fide autism gene like CHD8. These environmental risk factors have a less strong effect than the genes that we know about.
And at one point you mentioned there was one common variant that some people have that don't have autism.
Right. So the common variants in autism, this basically, we need tens of thousands of people to participate so we can understand this better. So far, researchers have only analyzed about 16,000 people with autism, and that's every person with autism that's basically ever participated in a research study, an autism research study, and they've identified one variant with a very, very weak effect on autism risk. So there's nothing, I don't even remember what chromosome it's on, but there's nothing clinically that you could do or you would know about with that common variant. It's a tough thing to crack, a tough nut to crack.
Can a parent be a carrier of an autism gene and not have autism themselves?
Yes. We're seeing this more and more. In Spark, we have cases of both, where the parent has autism or Asperger's or ASD diagnosis and passes on a variant to their child. And then we have cases where they're passed on from an unaffected parent.
Is there any research into motor coordination being linked to genes in individuals with autism?
No. I mean, I think that it's clear that motor delays, motor deficits are very prevalent in autism, and that people with these de novo changes of strong effect have higher motor symptoms than others, but there aren't specific genes that are known to have a specific motor effect.
Okay. This is sort of a high-level question, but I think we can answer it. Is Spark using an array-based or next-generation sequencing-based
That's a great question.
Analysis?
I love that question. We're doing both. So in Spark, the DNA is being analyzed with a microarray, a high-resolution microarray genotyping chip that looks at about 870,000 markers in the genome. And And we're also doing exome sequencing, which is next generation sequencing.
How about grandparents? Can they join Spark?
No, not now. We don't have a way for grandparents to join Spark. I think if a grandparent was a guardian. Oh, right. Or if it's a multi-generational family where there's one generation that has a child with autism and they themselves also have autism, then yes, they can invite their parents. So, yeah.
Okay, so once someone receives results, how can they use them? Would that help inform things in the future?
I think it depends on the actual results. I think some of the genes that we're returning, that there can be clinical implications. There's one gene on our list where the type of variant that you have can help inform epilepsy medication. I think it depends on I think it just is a very case-by-case basis, but I can say that there definitely have been cases where we've returned results and the family has been able to use that information to guide that individual's treatment.
Here's one that I'll just answer myself because
Sure.
I'll give a little more. So someone was asking how they can get more information, handouts, or brochures. So if you're interested in spreading the word, you can email us at info@sparkforautism.org, and we have materials on hand that we can send out to people. Can the father pass on the autism gene?
Yeah, we have cases of that too. That can happen. And in both ways too. Someone who's unaffected might have something, and for some reason they are not affected, and they pass that on to their child, and the child's affected. I think in these cases, what we've learned is that there are so many factors of autism, and so I think in these cases it's a mix of genetics and environment that contribute to the reason why the person with autism has autism.
So Spark has been running for a number of years now, and so someone is asking if they were an early participant and maybe their sample was sequenced and we didn't find anything. Will it be sequenced again? Might they receive results in the future?
Yes, definitely. It could happen. I think that basically what we're doing is an annual review of the data, so maybe even more often than that in reality. So, but I think that it's definitely possible for someone to get a result later on, because we're always growing, and as Spark grows, we're learning more and discovering more. And so every year we know more and know more genes. So even in this past 12 months, we're adding 20 genes to the list.
So even if someone gets a result and they can't use it clinically at the moment, are there benefits down the line?
Yeah, absolutely. So I think that no matter what, being able to understand the cause of autism and reach out to other families with this same condition, can be really powerful and really helpful. I think that having this information is really helpful because in the future we will have more targeted studies to particular diagnoses, and as the numbers grow, that will just increase. And so having that information, I think, is really powerful and could really be informative later on.
Can you talk a little bit about how many samples we have now at the lab? So, the progress of how we've been sequencing?
So we have 4,500 families on our sequencers right now that we expect the results, the data, by late May, so in a couple of weeks. I think that first data push is going to be really impactful, and from there we'll be able to return hundreds of results. We also have 2,500 more families in our pipeline, so about 7,500 total. And we're also sequencing people with just one parent as well in Spark.
How have some of the families that have received results already responded to the news?
I think that it's been really positive. I think that I've been on all of these calls myself, and I think that people feel a sense of relief, just understanding what it is. And, you know, it's mixed because some of them are older. Some of the individuals with autism are older, they're adults, but some of them are really young. And so I think that also plays a role in the response. But I would say it's been overwhelmingly positive.
Is there any research that points to a relation between genetics and, say, mental health conditions, ADHD or anxiety, depression?
Yeah. So the genetics of autism is there have been lots of comparisons to other mental health conditions. So the genes that are risk genes for autism also are risk genes for ADHD, schizophrenia, and bipolar disorder, less so for depression and anxiety. So people have actually looked at the similarities and the differences, and so we know that there's a lot of overlap with schizophrenia, bipolar, ADHD, but not as much with depression and anxiety.
Someone's asking a question about treatment benefits based on the results, and I recall there was a story that maybe someone was able to guide their treatment a little bit based on the results.
Yeah. Not medical treatments, but because you have an understanding of what the trajectory looks like, the developmental trajectory, because even that is hard to predict. When you have a young child with autism, you really don't have a lot of indicators of what's going to happen. But when you have a genetic diagnosis, you have a clearer picture of what you might expect. And I think even knowing that has helped.
All right. Looking through some more questions here. Feel free to chat any more in if you have them. I know this is hard to say, but is there sort of a timeline about how long it might take for someone to get results if we find them?
Yeah, I think now that we are definitely, now that we've really been operating for 2 years and we're on a more regular sequencing schedule, I would say, I think it's important to know that we definitely need to accumulate a lot of samples before we sequence, so we're sequencing in large batches. So it's not like we get your sample and we sequence it the next day. I think that the longest time from now going forward is probably about 6 months between the time they sign up and sequencing, but then it takes time to analyze the data, and that can take anywhere from 3 to 6 months, maybe longer, because we want to do things right and we want to make sure we don't return any false results to people. So there's a lot of quality control and a lot of checks along the way. So,
can you talk a little bit about why we need tens of thousands of people?
Yeah. So we need lots of people because of the heterogeneity of autism. Because there are so many things that contribute to autism, and so we've been talking about today how sometimes there can be an unaffected parent who has a variant that passes on that variant to the child, and that child has autism. When you have cases like that, where it's not always black and white, you need a lot of numbers to power your analyses to be able to say, most of the time when this person has this variant, they have autism. And it's just because of that, those are the two biggest reasons why we need a lot of people, because there's a lot of heterogeneity, because there's lots of genes that lead to autism. So you need to look at a lot of people to find all of them. And because of this, the genes, a lot of the genes aren't perfectly correlated with autism. So for some of these variants, you have people who have the variants but don't have autism. And when that's the case, you need a lot of people so you can be sure that what you're seeing is real.
Other than genetic studies, what kinds of research is Spark doing outside of genetics?
Oh, that's a great question. So what Spark is doing is we have this platform that we call Research Match. So we allow any researcher who's interested in recruiting people in Spark for their own studies. So last year, I think we had 10,000 people in Spark participate in these other studies, and they're really variable. So some of them are in-person studies, like intervention studies, looking at oxytocin, looking at, trying to think of the other intervention studies, looking at a behavioral intervention, so that kind of study. We also, there was a large study on environmental risk factors, so there was a scientist from Johns Hopkins who wanted to understand prenatal exposures in autism, so surveyed 2,000 autism moms to get a sense of what their pregnancy was like. We also had a large study of several thousand people. The researchers wanted to understand non-drug treatment patterns geographically throughout the United States. So just lots of different research and more to come in 2018.
Can adults with autism participate if their parents do not participate?
Yes, absolutely.
And can someone join Spark without providing DNA?
Yes, definitely. So you just, the first consent form, and then the other one you don't have to fill out.
Someone had a question about the kinds of analysis that we use, the kinds of genetic analysis that we use. Can you maybe explain a little bit more about that?
Okay.
They were specifically asking about machine learning.
Oh, machine learning. Okay. So the analyses that we use are pretty straightforward analyses where we're taking all the data and looking for things that are just present in the person with autism and not present in their parents. And that doesn't involve machine learning. It definitely involves bioinformaticians and lots of technical people helping and doing these analyses. We also spend a lot of time looking at these missense mutations, and that's definitely an area where machine learning would come into play. I think for the straightforward analyses, we're just looking for things that we can predict their function or the impact of, and that we don't need machine learning, but there are definitely more novel analyses that we're doing that will use machine learning and other kinds of AI.
You've mentioned this a couple times, but just to reiterate, is there clarity on the number of genes involved in autism?
It's somewhere between 500 and 1,000. That's not much clarity, but it's definitely more than we have now. And whether that number is closer to 500 or 1,000 is an empirical question.
See, if someone is thinking about ordering testing through their provider, would we use that data, or would you recommend that they also enroll in Spark?
I would recommend that they also enroll in Spark. We are the genotyping chip that we're doing. All that information is going to help power the analyses for those common variants. And if we don't have your data, then we can't. Then we'll have one less person. So that. I think there are definitely reasons why it could be helpful for someone, if they're willing, to participate.
Please keep sending in questions. This is really great conversation.
Oh, good.
where do you think autism genetics research will be in, say, 10 years?
Okay, that's a great question, and one that I can speculate on, so that's great. I can just make something up. In 10 years, I do think that we will have an understanding of all the really high-risk genes for autism. So the things that have a strong impact. We might not understand every tiny genetic change that can contribute to autism risk, like all those common variants, but at least for all the high-risk genes that are involved in autism, we will know them. I also think that there will, in 10 years, I do think that there will be some treatments for some subset of individuals with autism, whether it's people with a particular phenotype or that means trait, or particular genetic change. We don't know yet, but I definitely think there will be some kind of treatments available that will improve the core symptoms of autism in some subset of people. Might not be everybody, but it might. I think for a subset we'll be able to find something. I think if there's anything else. I think that the more that we understand about the genetics that lead to autism, the better accepted autism will be in the community. I think that there are a lot of people out there who still don't understand like how heritable and how genetic autism is, and so there are lots of other causes for autism that are discussed and talked about that don't have a lot of scientific value. So I think that the more that we understand about the genetics, the better off we will all be.
Someone was asking about how our samples are processed. Are they in a certain order or prioritized in any way?
No. So basically there is a lab in Wisconsin and they receive all of our samples. So their job is to open up tens of thousands of boxes and unpack them and extract the DNA, and they just take it as it comes. And then we do a lot of quality control on it. So a percentage of the samples fail. We just aren't able to get enough DNA out of them, and so when that happens, there's a process for automatically contacting the family member and asking for sending another kit and getting another sample, and then once we get that, it just goes into the queue. So it is sort of order-based, but it's also definitely prioritized for the complete families first, just because that's the best genetic analysis that we can do. If we have the parents and the person with autism, then we could, if there is a de novo change, we'll be able to find it. And those are the things that right now we have the most confidence in. That doesn't mean that if you don't have both parents that we're not going to analyze your DNA, because we definitely are, and we're already starting to sequence incomplete families and people without any family members in autism. So it's just, I would say that the results that we're able to return are definitely heavily weighted towards the de novo variants. But if you saw on the list, there were inherited variants also that we're returning. So, yeah.
Are we considering any other types of collection mechanisms, blood or
yes.
different
So
types
actually right now we're just about to launch a pilot for a different method of collection. It's basically a sponge that gets swabbed on the inside, and it's not as detailed as the current saliva collection kit. We're going to pilot this. I do think that it will work in people who really can't spit. I will say that because the amount of DNA that we can get out of the cheek swab is much less than we can from spitting, that there are also sort of issues with that, because we need DNA for analysis, but then if we find a result, we still need DNA. So there actually have been cases where we think, this is going to happen. Like we think we might have a result, but we need to confirm it, and so we have to reach it back out to the family and ask for more DNA. So that's the issue, the trade-off with the swab. But we're going to pilot it and see what we get.
If someone does get a result, what does that look like? How complete is it? What kind of information is included?
So what happens is that you'll get an email from Spark saying that you've got a result, and you have a choice of whether you want to get that result back from Spark directly or through your own provider. If you choose your own provider, we will fax over a clinical report that will basically say this is the genetic change, this is the inheritance pattern, this is the gene, here's some information about the gene, and that will all be transmitted to your provider, and you can look at that together. For people who get the result through us, a genetic counselor will contact them, set up an appointment. They'll talk to you over the phone and discuss your result with you. And you'll be able to ask them questions, and they'll be able to provide you some information on the change itself and the gene and any other questions that you might have.
What other kinds of results does Spark return?
So we just started returning some interesting results for some of the questionnaires that we ask. So there's one questionnaire that everybody in Spark fills out. It's called the Social Communication Questionnaire. What's interesting about our reports is that we show you the Spark average. So I think we've got 19,000 people who have filled this questionnaire out, 19,000 people with autism, and we show you where you fall in relationship to the rest of the cohort. And I think that could be useful information for a lot of people. So that's one, and we plan to do the same thing for some other questionnaires. There's a questionnaire on repetitive behaviors that will show you where you are compared to the rest of the cohort. And there's a motor coordination questionnaire. If you took that, then we'll be returning results on that.
How long will the Spark study continue?
It's indefinite, I would say. We're the foundation here that is our funder is definitely committed to Spark, and I would say the next 10, 20, 30 years.
Would Spark consider looking at epigenetic markers?
Yeah, I think that as technologies improve, there might be ways of looking at epigenetic markers in a scalable way. I think, yes, the field is very open.
Can you explain the partnership between clinical sites?
Sure. So we have a network of 25 different clinical sites across the country. So we ask clinical sites to apply, and there's an application process, and we chose 25 of everyone that applied, and they work directly with their patient communities and their local communities to recruit for Spark. So if you live near one, definitely contact them if you're interested. You don't have to go to the site directly to sign up. They can help you by phone, but if you wanted extra help, they're there and located all across the country. And so I've actually been able to visit a few of them in the past month, and it's been a really great experience to see all the on-the-ground work that goes into recruiting for our study.
Right. Oh, who is funding Spark?
So Spark is funded by the Simons Foundation Autism Research Initiative. So the Safari, you can go to the website at sfari.org, is a privately funded organization that funds autism research in the United States.
All right. Well, questions are kind of slowing down, so I think we'll end just a few minutes early. Thank you so much, Pam. This was a great presentation, and thank you to everyone that asked questions. We're happy to be able to talk to you all. We don't always get this opportunity. We also want to give a shout out to our Community Advisory Council, who helped to advise an earlier version of this presentation, and we took everyone's suggested changes and feedback into account, and ultimately we're able to produce a really great presentation for you all. So thank you again. We really appreciate you taking the time to listen to us.